Going. Welcome everybody to our webinar sponsored by our project VacCAP, Vaccinium Cap. We have a wonderful theme for this webinar today on exploring firmness and texture in blueberry and cranberry. My name is Amaya Tucha and I'm part of the extension team of VacCAP, and I'll be the moderator for this webinar. So before I introduce the speakers for today, one of the things that I want to go through is just some housekeeping issues. First of all, please make sure that you stay muted so that you don't interrupt the speakers. If you have any questions, please feel free to just type them in the chat box, and we'll address all of them at the end of the webinar so that we don't interrupt anybody. And we will have at the end of this webinar a survey that will be distributed, and we would highly appreciate if you could answer them, and you're going to get that through an email. So with that, I'll just go ahead and introduce our great speakers today. We have 3 speakers. This webinar is going to consist of 3 small talks. And the first speaker that we will have today is Dr. Penelope Perkins-Veazie. And she is a post-harvest physiology with North Carolina State University. And her talk will be on the introduction to exploring firmness and textures in blueberry and cranberry. Then we will have Dr. Juan Zalapa, who is a USDA-ARS research geneticist and a professor at the Department of Horticulture at UW-Madison, and his talk will be about measuring cranberry fruit quality and firmness. And then our third and last speaker will be Dr. Lara Jongo, that leads the berry genetics and breeding programs at the Edmund Mach Foundation in Trentino, Italy. And her talk will be about texture methods and applications in blueberries. And with that, I will pass it on to Dr. Perkins-Veazie to start her presentation.
Okay, great. Hold on a minute here. And presentation. Okay. Okay, so good morning, everybody. And there's a little bit of things here. Okay, so yes, I'm going to do the overview here of kind of introduction overview of textures so that people have a little bit of an idea of what we're trying to get at in these talks with blueberry and cranberry. I'm located at North Carolina State with the Plants for Human Health Institute. And this is to try and go a little bit over the instrumentation that's used along with texture, what is texture itself. So texture is related to the feeling of food in the mouth. And it has a lot of attributes that can be measured, both with instruments and sensory. And these include things like firmness, toughness, springiness, etc. The problem we have is how do we make this fit? The texture analyzing really started with inert objects like rubber. Now we've moved it into foods, and now we're trying to make it some sort of quantitative application that we can apply at will. And usually these come down to what people have studied over the years as they build firm testers, looking at firmness first, and that's the measure of a yield of a surface to a force. Puncture is resistance to perforation or piercing. Very important if you're looking at how a berry would stand up to being punctured by a stem, for instance. Elasticity, or Young's modulus, is a relatively new concept in the fruit world, and it's a measure of stiffness and the ability to recover from deformation. The difference between this and springiness, which is also a type of elasticity, are what you use to get to those ratios. One is independent of shape and size, and the other is dependent on shape and the amount of strain in the material. And this is a little bit of what you see here. Here's your stress, here's your strain, here's your elasticity of your crop or your berry. And the reason I bring out springiness and elasticity, you could have very round blueberries, or you could have some slightly flattened blueberries, and this could change these 2 values. So that's one reason that both are looked at. The other thing that we look at a lot is how do we estimate texture? A lot of people ask, what should I be buying? What should I be using for a texture instrument? And really, ultimately, if you talk to the food science world, they're going to say sensory tests are the best way to go, which they are. They're the gold standard. The problem is for a breeder or for people who have particular studies, these can be very awkward to set up. You have to have your panel. They can be quite costly because many times you have to have 10 or more people. They take a lot of time to set up, and also you have problems with logistics, trying to get your berries out of the cooler, for instance, and comparing them against fresh berries can be very challenging. The other problem that comes up many times is if you're walking through a field trying to evaluate your selections for firmness, that's really tough to do. You want something that's going to be better than your finger and thumb, because by 2 hours in, your finger and thumb are pretty tired. But how do you do this with all of these hundreds of fruit? You certainly can't do it with a sensory test. And then the other problem that we've run into, especially in the past on blackberries, is that you had a very firm fruit in the field, but it becomes very soft in storage. And so then your correlation of what happened in the field does not necessarily correlate with what happens in storage. And then the other is, can we try and find an analytical test that would mimic sensory, that would become the screening gateway? So you can start with your hundreds of fruit samples, do quantitative value measurements, establish what your quantitative values are that you want, and then start doing the sensory work to make sure that those are in target with what consumers really want. So why are we looking at texture? Well, there's some very practical reasons for that. One is resistance. These are harvesting issues, resistance to bruising in particular, as we try and move more into machine harvesting systems. Harvest temperature, all of these machine harvesting, and also the drops that happen in a packing line will all cause bruising effects. And there may be small bruises and there may be cumulative bruising. The other thing that happens is, skip down here to resistance to injury, punctures that can happen during the harvest and handling systems. The others are when you go into resistance to damage during transport. If you have a semi-load of berries, even though they're well contained in a clamshell, they'll still be up and down movement and all around movement. You hit a bump and the whole load will go up and down. And you do everything you can to mitigate this, but you can still damage your fruit. And then I'm going to go back up here to post-harvest changes. So ultimately, you've done everything right on your harvest and handling system, but for whatever reason, you may have other problems post-harvest. You may have excessive water loss. You may have mealiness that's coming up. The berries look fine, but somebody bites into them and they don't like the way they crunch. They have loss of cell wall integrity. They may be soft, mushy, or they may feel like you have a bunch of starch grains in your mouth, which is mealiness. So we want some way to get a handle on this and figure out what's causing it or how we can keep it from happening. And then finally, also looking at consumer preferences. We have crisp types of blueberry now. Crisp has become something that we used to consider soft fruits. We now have crunchy or crisp types in some of the newer varieties. So again, we need to distinguish between what consumers want, which might be crisp and it might be melting. And we have to be able to put a number on that and ultimately a sensory test on that. So what are the types of instruments used for firmness? They can be relatively inexpensive. The handheld instruments generally are much less. You're talking anywhere from $100 to $500 usually for these. The McCormick puncture resistant tester has been around for 100 years. It's first used on apples. It's used on corn. The VacCAP was one of the earlier inventions, but these are a simple dial thing. The problem is they're very simple to use. The problem is you have to have the same person taking the reading. Otherwise, your reproducibility goes down to about 50%. So that's always the problem with these systems. They're great for kind of a guide as to what you're looking for, but not an absolute. And the other problem is they can be very tiring to use. If you've been working with these for 10 hours in the field, you're pretty exhausted trying to read the dial, capture everything on there, and keep piercing with one hand. There's some other types out there. The Tironi fruit firm seems to be a considerably easier to use and probably a lot more ergonomic. I've never tried this, but I've been interested to read in this. This seems to be getting pretty decent values for resistance to deformation. The backslow spring force is another popular one. And this has been used, I know it's been used with blueberries. And the idea is you put your blueberry between it and you get a reading here on the dial. It doesn't stop. As far as I can tell, it never stops on the actual maximum value. So you have to keep your eye on this one. And then one that we had done many years ago was we adapted a Wagner force gauge. So we were interested in the resistance to puncture in a raspberry. So we fitted an insect pin on the end of it. And we never went many places with it until spotted wing Drosophila came up. And it turns out that this is really useful for measuring resistance to spotted wing Drosophila. Mounted firmness testers are much more compatible with giving automated ideas of what you have, doing it much more rapidly, and it takes out a lot of the user problems. So except for maybe this one. But these Fruit Firm 1000 and the Fruit Tech both operate on the principle that you circulate your berries, so you can do this quite rapidly. The Fruit Tech uses a deflection system. It's a little compression. It basically gives it a little pop on the berry and measures the resistance to deflection or compression. The Firm Tech looks like it does it twice, so you do double compression. I haven't used this one yet. And then there's the other one over here. Let's see if I can. This is the McCormick, basically mounted onto a simple drill to press system. This gets rid of a lot of your your labor issues. You still have some where you have to pretty much regulate how fast you're going down with the drill press handle and making sure you don't go too deeply. And then ultimately the gold standard now is this texture analyzer or an Instron, where you can everything, there's software involved. You can automate the speed, you can automate the penetration, you can change your probe, and you can look at a number of variables on the software itself to figure out your elasticity and shear, etc. And of course, these cost quite a bit more. These instruments, the texture analyzer, Instron, can be anywhere from $20,000 to $100,000. Fruit Firm Techs are running, I think, around right around 8 to 10,000. So you have some options here. You just kind of have to figure out what works for your system. The types of tests that are normally done for these berries and have been looked at quite a bit, especially on blueberry, not so much on cranberry. Compression, which is what causes deformation, and then puncture, which is the degree of penetration. And then puncture can be, the width of the probe can change. Also with compression, you can have a very small plate or a very big plate. Puncture might be needle probe all the way to something that's maybe a ½ an inch in diameter. By far the most common one that's been used over time has been maximum resistance force. How much resistance to force does this berry have? As kind of a gauge of how it will hold up, for instance, for machine harvesting. What's becoming more common now is we have more sophisticated instrumentation and understand more about texture, is looking at the modulus elasticity. And then some of the other ones, such as gumminess and chewiness. These are more, I've always associated these more with food products, but you can now apply some of these to fruits coming out as well. And then cohesiveness. So what are the goals here? Really, we're going to look at one presentation on cranberry, where really cranberry texture is kind of a new area. And the other will be talking more in depth about what's going on with blueberry. And the idea is, can we move from the simple puncture and compression tests that are being used, that instead we might be able to develop more aspects that will give more information about the changes in the fruit that we can link back more successfully to post-harvest changes in quality. And then finally, can we attempt to use these values that we've developed and relate them to sensory values so that we have kind of a nice gateway so that we don't have to use sensory for everything. And then the final slide I just want to put in there, is just a shout out to the VacCAPs that's funding this project, fruit characteristics and fruit quality, and how these two are related with texture. And that's all I have. And I'll stop share. Okay. And let's see. Okay. It looks like we moved into Dr. Zalapa's here.
I was just testing it, yes.
Oh, okay.
Juan, you can go ahead and start your presentation. Thank you, Penny.
Thank you, Penny. Thank you so much for allowing me to give this presentation to the VacCAP team and everyone here for coming to listen. And I apologize in advance if I start coughing. I've been dealing with a cough that doesn't go away, but I took some medicine right now, so hopefully it will be fine. And it's not COVID, but I don't know what it is, so here I am. So again, the name of my talk is Measuring Cranberry Fruit Quality and Firmness. And I'm Juan Zalapa, and I, thank you so much for the support. I have a bunch of students that have done the work here, so I want to give them thanks, and the team for the VacCAP is, the Cranberry team, there's everybody's working on it, but the main people working on this is me and Amaya and Nick Borsa, Paula Ashok, Lara Jongo, and Massimo Yorizo, among many other people. And of course, Hector Lopez Moreno and Matt Phillips, Eric Wiseman. I have a problem changing the slide. Let's see. Okay, so the main emphasis for our research is to develop techniques that can be useful for development of sweetened and dried cranberry. So things that we're looking for is uniform color, uniform size, and fleshiness. We are very interested in. Okay, sorry, I'm not able to change.
Juan, in the lower left corner of your screen, you probably will have 2 arrows that will allow you to advance.
Okay, yes. All right, that actually is because my regular arrow is not changing. So these are the main areas that we are interested for sweet and dry cranberry production: color, size, and then fleshiness in the inside, and of course firmness. And so we developed a project to measure firmness, which is one of the main things that we're interested for cranberry or dry cranberry production. And we use this machine, which is already talked about by Penny. There's really not much literature on how to measure cranberry firmness, and also the cranberries are very different anatomy, as related to other fruits. And so we decided to study, besides firmness, other quality traits that maybe influence firmness. And our goal was to define the test that works good for cranberry using this machine and other techniques. And so some of the techniques that we are using is looking at the internal structure. This is what I was saying, that in cranberries are slightly different inside. When you cut them, they have the locules. And so we believe that the locules affect the firmness of cranberry. So we started playing around with cranberries and came up with this idea of just looking at the internal structure. And cranberries, as they grow, they fill in from the inside. So here's a small cranberry growing, and then as it grows, it just fills in more flesh. And we believe that has to do with the increase of firmness over time in cranberry. So we developed this stamping technique. We're looking of ways of not doing the stamping and actually maybe doing some computer scanning, because that might be easier. The stamping takes a long time, but we also develop a software that we can look at the inside and calculate the amount of flesh, the amount of area around the outside and then the inside, and all kinds of different measurements that we can look at. We can also look at fruit size from inside this way, and together with the cranberry external appearance, we basically just take a photo, and then this photo can give us ideas of also the size, the shape, and color. Color is very important in cranberry, and how all these things are related to firmness. We know that cranberries develop over time. They develop the firmness as they grow. So, and then also color is developing at the same time as firmness is developed. So at some point they go together. So we've collected a ton of data over the last few years in the VacCAP project. We've had some challenges with the coronavirus, but overall we have 3 years of data for cultivars, for 10 cultivars, and then we have 3 biparental segregating populations, a total of almost 500 clones for 2 years. This is all the data that we have to this point. We have 71 fruit quality traits overall. 13 traits are external appearance, and then 11 structural traits inside, and then the firmness are 47 traits. I'm going to talk about a little bit about the cultivars. So we have data on 10 cultivars. Here I'm presenting an analysis for one of the years, and what we did is we have 3 years of data here, and one of the years we collected all of the cultivars at the same time. The collection time was at the same time, and then after we did that first analysis, we realized that maybe we needed to collect the cultivars at the best harvest time. So the second 2 years we did it at 3 different times. And so I'm presenting here the analysis for the year where we collected all of the cultivars at the same time. And in the future, I will present a comparison between year 1 and year 2 and 3. So our goal is to, again, detect differences in firmness in known cultivars of known firmness and storage differences. So in this case, we are comparing basically 3 different types of cultivars. The ones in red are firm, very firm, and then the ones in the middle are more middle firmness, and then the ones in the blue square, they will be more soft. And so we have 4 storage times, 10 cultivars, and we have a total of 40 comparisons where after the storing 4 times and the 10 cultivars, that gives you 40 different comparisons that we are going to be looking at here. So for firmness, again, we have 47 traits, and I'm going to show some of the results for these 47 traits. The main thing is that, you know, again, we have 40 different comparisons if you account for the 10 cultivars and the 4 storage times. Some of the tests are able to detect those differences more readily. So, for example, maximum force is the standard way that people usually look at firmness, and that can detect 23 differences, and some can detect 25 and 26. So we're looking for the test that can detect the most number of differences among the cultivars and the storage times. So here is a PCA. This took me a while to understand, and I'm still trying to like get a lot of information out of here, but this is a PCA where you can see the cultivars that are firm in our study. They ended up on the right side of the plot. And then the cultivars that are more soft, they tend to be more on the right side of the plot, or on the left side of the plot. And then the right are the firm ones. So, you know, this plot uses all of the data from all of the 47 firmness tests and allows those cultivars to separate actually the way that we were expecting in terms of firmness. So each of these lines represents all of each of the 47 traits that we measure. If the lines are very close together, that means that the tests are producing similar results. You can see that a lot of the double compression tests are kind of grouping together. That is the tests that are related to one double compression basically measurement. Then we have the puncture, the blade, and puncture is in blue, blade is in green, and then the Kramer test is in red. So this allows us to see that our data is separating the cultivars, and we can also see which tests are more similar, and then we can start choosing tests that are giving us information that is more different. So I might want to choose maybe 2 of the tests in the orange, and maybe a green test and a blue test and a red, and I'm trying to cover the variation. Overall, all of the tests together, you know, didn't necessarily account for a ton of variation. So for example, the highest trait for the dimension 1 was W2 and only accounted for 3% of the variation. On dimension 2, we have 11% for SP2. So, you know, this test, you know, we're thinking about a way to maybe using, since we have the double compression test, maybe there's a way that we can use all of the tests together to give more separation. So we're looking into that and see how we can maybe use all of the tests together. And you know, we already have the data, so maybe there's a way we can use it all together. But other things that we're looking at is the average coefficient of variation. We're looking for tests that don't have a lot of variation. So here, some of the ones in yellow have the lower variation. This is impacted a lot by sample size. So we did some tests to see what is the appropriate sample size. We believe that, you know, for cranberry, maybe we need to be at least in 30 samples, 50, 75 would be better, but it's really challenging to get to over 50 samples. I think at this point, we're doing maybe around 30 or 50. So we definitely can get past the 100, but the sample size is important. About 100, you know, samples, it probably doesn't make too much difference anymore. But, you know, we're somewhere in the 50 range. Other things that are interesting is the correlation with fruit size and with color. So the traits that are more correlated with fruit size, and those are, I believe, maybe less useful or maybe somewhat biased. So, here is, for example, a fruit size correlation with width, length, and area. You can see the correlation very clearly with maximum force. So this is the standard measurement that people use. But if you take, another measurement, like for example, stress strain area that takes into account fruit size, then the correlation kind of seems to be lower. In this case, it's a little bit negative, but it's in terms of the magnitude of the correlation is a lot lower. Also, you can see that the cultivars that are the firm cultivars, they have a trend. So you have the blue are the cultivars that are more firm, and then the orange are the medium, and then the yellow are the, so this makes sense that there will be a correlation. We did conduct a correlation with all of the traits that are for the cultivar. So we have the internal structured, and then the external appearance, and then all of the 47 firmness traits. And so this table is very complicated, but just to say that, fruit size, a lot of the fruit size measurements are correlated with maximum force and with some of the other firmness measurements, and also color. So, we're, trying to think about how to use this and what is the best way of measuring firmness that is not so correlated to fruit size. And perhaps we need to use several of these tests and then see which ones are useful. Here's just a little bit of separation. These are the 40 different comparisons. So you have in red the cultivars and the red at the fresh time, and then this brown is the same cultivar at period of time after storage, and so on and so forth. So you have 4 different storage times, and overall we can separate the cultivars. Here we're not showing the separation at the fresh at the same stage, but there's separation. I'm just showing here the separation for the storage times, and very easily we can see the separation and storage times. So we can also see the separation across genotypes at each of the storage times. So this is maximum force. This is the one that is correlated to highly correlated to fruit size. And this is double compression stress strain area. And this one is less correlated, or slightly negative correlated in this case. But so we can use, what our goal is maybe to use both of these is part of the double compression analysis anyways. So we can easily use both of this in downstream analysis for the QTL analysis, and see really which one allows us to find more QTLs and do better breeding decisions. Here's puncture, very similar results. And here I'm showing some of the most promising firmness techniques. And blade work, for us, blade work didn't do much. It didn't separate the cultivars, it didn't separate even the storage times. In some cases it did, but overall it was pretty hit and miss. Here's maximum force and blade, and the other one was work. And we're also interested in looking at frozen versus fresh. So the red is the separation of cultivars in fresh, and then the blue is after freezing. So overall, no matter which technique we use, we can't really find any separation after freezing. But in reality, a lot of the fruit in cranberries are used in frozen for the technique of developing dried cranberries. So we're looking at techniques that allow us to separate frozen. Overall, the difference between fresh and frozen is like a 10-fold difference. So this is maximum force, this is stress strain area, and this is puncture. Again, all of the fresh techniques separate the cultivars, but not so much in the frozen. So for that purpose, we're doing some tests with the Kramer Seer test. So this is a test that uses basically a box. You put a bunch of cranberries in there, and the probe just presses on them. In this case, this probe is doesn't have a lot of sharpness in the blades, and so we're developing a probe that has sharp blades. And also we're thinking that maybe a puncture probe might work well for this. And so this is our next test, and we're going to do this in fresh and after freezing and see if we can develop a technique that works better. That said, I also have data for the mapping population. So in this case, we have data that is only in one harvesting time. Again, this could be a problem. Maybe we need to have more harvesting times, but right now this is what we have is 2 years of one harvesting time. We have a bunch of correlations also for that. I'm not gonna go for them, just to say that some of the correlations that we saw in the cultivars did not hold once we look at the population. So we need to think about why that is. Perhaps is you know, having so many cultivars and only one harvesting time, we're not allowing the cultivars to, or the genotypes to acquire maximum firmness. So we have to see why some of those correlations didn't hold up in this study. And then, you know, the plan is to use all of that for QTL analysis. And this is just a preview of what is coming. So things that I like to discuss maybe at one point, and I want some of you to think about, is ideas for the firmness in frozen. I mean, this is how actually the cranberries are processed. They're frozen and then they're taken out of the freezer, and then they're sliced after they have been frozen. And so what is the you know how we can develop a technique that is more closely to the industry. And so really, what is the relationship between the type of testing that we're doing here and the actual production? Because this is you know, not exactly getting at that. And then finally, the correlation between fruit and size and color, you know, how we can deal with that. Does it matter? You know, if there's other experts here and people that have ideas, I'd be happy to talk about this. So with that, thank you. And thank you to the students that did all the work. Thank you, Amaya, Nick, Massimo, Lara, and everyone else that I might forget. Thank you so much.
Thank you, Juan, for your presentation. And then we're going to move on to our last presenter, Dr. Lara Jongo. And after that, we will open up for some questions. I know that there's some that have been already typed into the chat. So if you have questions, just go ahead and I'll read them to the different speakers. So just ask your questions to the chat. Thank you.
Thank you. Hi, everybody. Good morning or good very morning on the other side. My name is Lara Jongo. I work at Edmund Mach Foundation, as Amaya said, in Northern Italy. And I'm mainly involved in raspberry and blueberry breeding and fruit quality. And this is also the topic we cover within the project VacCAP. And I really need to thank all the people working in this project because it is a very generous community and very fruitful. So as Penny said, when we talk about texture, this is a matter of interpretation, first of all, since texture is a complex sensory property, which is a multi-parameter attribute. So many parameters contribute to the dissection of texture that depends on the structure of the food matrix we study and is detected by several senses, sensorily speaking. Consumers estimate the firmness on the basis of the deformation caused by a physical pressure, and this may be ascertained by using the finger or hand or by biting and chewing on a product. And the same happens for blueberry, but it is quite subjective, first of all, as also Penny said, and especially if you need to select for this track into a breeding program is not really the most interesting way of doing it. So we try to find a good compromise, choosing mechanical properties that have a physical meaning and that can be quantified in physical units, trying to make objective data out of this complex trait. So what do we need to consider if we aim to dissect blueberry texture? So saying dissect, we need to understand those that are the parameters that really mean something and that are able, as Juan said, to differentiate the cultivars and give us information that we need. So we need to consider the metrics we work on, the geometry of the fruit, the anatomy of the fruit, the histology, so how the tissue layers are into the blueberry. The genetic diversity, the gene pool we work with, because it can be a quite narrow gene pool or it can be a very wide gene pool. The environment that is applied to the development of the fruit or at harvest and in post-harvest of this fruit. Then we need to consider the protocol settings. So those parameters that are really significant to tell us a story on those berries. So we need to filter and to find the points that are really robust in discriminating over the different curves. But these curves can be different according to the test we choose, or to the probe we choose, or the speed test, or the method. So there is a study design that has really to be taken into account approaching this technique. And then when we have something in our hands, we need to standardize in order to get the technique standardized over equipments, so there are different machines, different labs, and different operators. Texture composition depends, as I said, on a hierarchical order and interaction of molecules, cell structure, cell-to-cell adhesion, tissue types, and tissues composing this final fruit anatomy. And during the development and then in post-harvest, cell walls remodel, cells and tissues change, and the cuticle modifies. So we need to take into account 4 major properties considering texture. The mechanical ones, so those that help us naming and labeling a fruit: firm, hard, chewy, fracturable, sticky, dense. Geometrical properties, so that a fruit can be, a blueberry can be smooth, grainy, fibrous. Then we can consider moisture properties, so a blueberry can be juicy. And last but not least, acoustic properties, because a blueberry is better if it is crispy or crunchy, whatever we want to call it. So within the VacCAP project, 2 years ago, we have started an experiment in order to compare 3 different methodics, 3 different methods on texture analysis in order to evaluate the discriminative power of the methods. The first one was a penetration with a flat probe. The probe was of different diameter. Here at FEM was chosen a 4 millimeter probe, and at North Carolina University a 2 millimeters probe. Then we set up the methodology for a needle puncture with a needle of 1.8 millimeters and a double compression TPA. We started to design, to tailor the methods over 24 accessions, 12 known cultivars that were, some of them were in common at FEM and in North Carolina, and 12 that were from our breeding program, and included northern, southern, high bush blueberries, and also hybrids. We measure the fruits at harvest and in post-harvest after 6 weeks at 2 degrees, 95% relative humidity. And as you can see here, you can already see that the curves that the analysis perform are really different. So our first step was to look at the curve and get the best parameters. And related to the penetration flat probe method, this was quite useful when an overall evaluation of the texture of both skin and pulp in a kind of static way was needed. Variation into our cultivar set was quite high, and also with a very limited number of parameters, which confirmed our previous publications. So we used the maximal force, the deformation, and the apparent Young modulus to express the variation within this gene pool. Originally, and this just to tell you how important it is to filter the parameters, we had identified 34 significant points over the curve, and we came up just with 3 that were really robust. The second method is a needle puncture, and this specifically designed to be sensitive to describe the skin of the blueberry. And this clearly differentiates the skin tissue layers in blueberry. What happened was that we were able to identify, in particular, 4 different points over the curves in these 24 accessions that we named epithelial force. You can see it on the right of the slide. Epithelial force that happened between 2 and 3.7 micron. Into the depth of the blueberry, hypodermis force at 0.24 and parenchymatic force at 0.34, while at 1.53 millimeters we consider it already inner forces contributing to this definition of texture. What was interesting was that the methodology was able to dissect the 2 more close forces, which were the epidermal and the hypodermis forces. And as you can see from the distribution of the accessions, they are orthogonal, so they are independent. And so we can understand what is the contribution of the 2 different tissues into different plant fruit materials. So this is a new method that enables a very fine assessment of the texture of blueberry skin components. And it is very important because if the penetration with a flat probe can be considered like a general information on the texture analysis, in this case, we go deeper. So being the skin, the tissue where the majority of the healthy bioactives and the first barrier to pathogens and insects, this is really an important information to be known of our genotypes. And this within the project was and is implemented in the Revealed by Arlen mapping population by the North Carolina State University. The third method was the double compression TPA, which is widely used in many matrices. It is a test that can predict how much each parameter can be retained after a first compression. And it can be useful to describe indexes like springiness, chewiness, resilience, hardness of the fruit. So to give a different vocabulary to call our metrics. So here the distribution of our genotypes changed again. So we had other additional information on the same gene pool that we were analyzing. In particular, with this technique, we saw that the loading vectors that had more value in this separation were hardness and work. Hardness is, in this case, the maximal hardness that corresponds to the force needed to compress 20% of the berry diameter in our case, while the work is the energy needed to produce that level of compression. While SF2, which means the second slope, so the ascending part of the curve in the second cycle of compression is contributing for the most as a vector into for the second factor, for the second component of the PCA. In this case, for example, this is quite clear. This selection was before absolutely interspersed into the central cloud of the varieties, while now it can be characterized, in particular for the slope at the second cycle from the other ones. So these are all information that add the knowledge to us of the genotypes we deal with. The TPA double compression test, as I said, can predict how much each parameter can be retained. And these mainly are dimensional indexes distribute in a similar but at the same time different way. Again, our selections. The higher loadings were observed for chewiness together with hardness. In this case, we also evaluate what was in the literature, and in agreement with Pons, gumminess and chewiness are mutually exclusive, and for blueberry, chewiness is absolutely more robust as an index to be used. Together with springiness, that in the literature is usually used in being calculated in two ways. So the first one as a ratio, and the second one just as the distance calculated over the second slope of the cycle of compression. In this case, so with this gimple, we used the D2, so just the distance didn't show any variation among the accessions, so that we are going to use the ratio. We need to consider that TPA is useful for simulating the force of biting or chewing on the fruit, imitating squeezing a berry during operations such as hand picking or consecutive dropping, and that might occur during mechanically sorted fruit. Related to bruising, susceptibility, post-harvest developments, and would also infer tolerance to pest and disease mechanism. So this additional information can help us also to relate to these kind of events. The second step was to move and see if we were able to get information also of the fate of these genotypes into post-harvest and how to measure the post-harvest changes we were seeing. Of course, we need to consider that the genotypes have different ethylene production and they lost water in a different way. So here we just described the texture evaluation. This is something that Penny also mentioned at the beginning, and if we have a look at a single point measurement, like can be firmness, for example, going from harvest, that is the blue point, to post-harvest, that is the red, we can see that there is a huge variation in the gene pool. So there can be very low values for firmness in among the genotypes, or very high. But then also in post-harvest, we have a very different patterns. So we have, for example, here quite high for firmness genotypes that remain very stable, otherwise that deploy very, very quickly, and other that increase the firmness from harvest to post-harvest. So we know the storage performance is genotype specific, and this can of course affect in the production pipeline, in the post-harvest management of blueberries, in particular during shipping and storage. So we can see that if we have a look through the use of the storage index, which is a logarithmic ratio of the individual parameters, how can they change from harvest to post-harvest, we can see that they can have, the different varieties can have a different profile if taken individually. So there can be a variety like Snow Chaser that increases, let's say, the storage index, has a storage index that is positive. Otherwise, that are more stable from harvest to post-harvest for the single parameters. This allows us also to get a distribution that is the composition not only of a single point measurement, like it might have been for firmness, for example, but a distribution for all the components we are evaluating. So the dissection of texture profiles might identify blueberry genotypes that do not only remain stable after storage, but some that also might improve after cold storage. And all these data are published in Post-Harvest Biology and Technology in a recent publication. Within the project, we also compared, as I said, with the aim to standardize these procedures, these protocols, two texture analyzers on the same cultivar set. There were a lot of variables. The sampling was done differently. There were different operative settings. The probe was slightly different, as I said. The environment, of course, was different. And this affected the correlation data in terms of lowering down those that were significant. However, we saw among the individual parameters significant correlations for most of the parameters into the three different methods that we developed. So they can be considered robust, and although other experiments are needed, they seem to be standardizable. TPA was the more robust among the three techniques in terms of correlation. Within the project at the North Carolina State University, they also started to automatize the evaluation of the texture parameters together with other parameters in order to infer some shelf life description and information related to the correlation with wrinkled or shriveled fruit. After meeting the advisory panel of the project, we were asked also to correlate the data that we were producing with the texture analysis with more easy and cheap handheld tools. So we published the data related to the fruit firma within the same publication, and there are strong correlation, although they are, let's say, correlated with most of the parameters without giving a strong distinction of that might be speculative into mirroring the different cell layers. And an ongoing validation is also, we are also doing a validation on other than the fruit firma. We introduced, as Penny said, as Dr. Perkins said, the fruit firma 1000, the Baxlo, the fruit hardness tester. The first preliminary evaluation we made are indicated here over a bunch of old, I would say, varieties of blueberry. And what you see is that being the value different, of course, for the three different methods, we see that the profile of the curve, if we look at them as a curve, although it is not super correct, but visually it is more interesting to look at it in this way, except for Duke, that for the fruit, with the fruit hardness tester seemed not to be standardized as it was with the other two testers. But we will be giving you more information on this as soon as the results will be ready. So what are the advantages and application of this know-how related to texture? As I said, we can have a very precise phenotypic characterization of the different genotypes that can be implemented with the different methods used. We can use the parameters as biomarkers per se, using them in a more targeted selection approach in blueberry breeding. We can have a better genotypic management and cultivar choice. We are evaluating if, in particular, the contribution of the skin of blueberry has a relationship with, for example, the tolerance in brackets to SWD. We published a work on grape that related the ovoposition of Drosophila Suzuki and related it to the three main parameters of apparent young modulus, maximum force, and force deformation into grape. So this is another work that we are doing in blueberry. So better genotypic prediction for post-harvest durability, again, linked to the skin and both epithelial, hypodermis, tissue layers. We can probably control better the post-harvest fate of our blueberries in terms of management, but also in terms of prediction. This is what we have started to do within VacCAP, in particular, linking the parameters of texture with the evolution in terms of mold and in terms of shriveling, hopefully associating this evaluation and this data to genomic information. So new parameters that can be applied for marker association studies and candidate genes that are texture related. We can also use the parameters as a more texture targeted approach for consumers and segmentation. So we can distinguish since the first evaluations among those fruit that can go to processing for the fresh market with a short or a long shelf life. More texture targeted approach for consumers and segmentation in terms, for example, of ability to distinguish between crispy, non-crispy, and so on. And the plan for our next future within the VacCAP project in particular is to finish the methodology on the acoustic parameters that we have started, aim at better phenotyping, crispness and crunchiness, and to evaluate better the post-harvest storage and texture dynamics, both in control and modified atmosphere. So I really need to thank my group here at Edmund Mach Foundation and all the team of VacCAP, in particular North Carolina State University team. Thank you.
Thank you so much, Lara, for your presentation. And I'm just going to pull up here our final slide. And before we go to any questions that the audience might have for our speakers, I just pasted on the chat a lot of links between them. There's a link to our YouTube channel for VacCAP, and we will have the recordings of that webinar there if you want to review some information. I also want to remind everybody that you will be receiving an email with a survey, and that survey, as Massimo was commenting on the chat box, is really important because it will allow us to get more information about your willingness to participate in certain workshops related with some of the methodology that has been presented today. And so with that, we will be happy to help answer any questions, and if there's any specific questions about the information that was presented today, you can just go ahead and type it in the chat. We still have about 2 more minutes before we end the webinar. So please go ahead and just type any questions that you have for our speakers. As part of the links there that I share with you in the chat, there's also the link to our website. We have plenty of information there on our website, and we also have the link to sign up for our newsletter if you want to learn more about other aspects of the project. So we have one question here. It's a specific question for Lara, and says it's very interesting and congratulations for a good job. It says, actually, in the packing house, the interest about firmness, crispness, and other texture information is very high in order to answer the quality requests of consumers.
Yeah, I know that that it is okay, but there are a lot of. She didn't finish to write.
Yeah, it says, but there are a lot of problems to measure this parameter by vision technologies.
Are they also allowed to, Amaya, to ask as a discussion, as an open discussion, or just?
We can do that too.
on?
If you think that that will facilitate answering some of the questions from the audience.
I guess it's just a matter of time, because if we have little time.
So I think that the question.
Yeah, I think the vision technologies. What I think is that is a good correlation also with the vision technologies. I mean, we need to work more on the correlation of the different parameters that are able to dissect the pulp to the skin and correlate with what we can get from an image analysis, both of the external fruit then of the internal fruit and the flesh.
There's another question here in the chat from Jessica that says, is there any reason why FirmTech 2 wouldn't give similar results for double compression measurement by running the same berry twice? I don't know if that question is for you, Lara, or for Juan. For Lara or for Penny?
I guess from my side, FirmTech 2 is a little less stable than the texture analyzer that we use, but at the same time has a lot of benefits and advantages that is automatized. I guess that the best way is to run a few analysis with a texture analyzer and correlate the data in order to see if there is a weakness on some of the parameters that you want to monitor, Jessica.
Yeah, I'll chime in here too. We tried this many years ago, Jessica. And what we were doing at that time was trying to see if the FirmTech would distinguish between varieties we felt were firm and those that were soft. And what we would do is move the berry instead of compressing it in the same place twice. We moved it a little bit, basically changed it around to the other side. We were not able to find that helped at all. And in fact, it seemed to make matters worse as far as segregating something that we felt phenotypically was soft and firm.
And for Cranberry, it didn't work so well either. Just, you know, I just don't know that it has, I couldn't, you know, I didn't own the machine. And just what I tried is didn't seem like it could adjust the amount of force that you could add. And Cranberries are slightly harder. So I just didn't feel like it was pressing it hard enough. So, but I didn't try too much, but it didn't seem like it worked so well for Cranberry.
Perfect. Well, we are 3 minutes past the hour, so I just want to really thank our speakers for sharing all this information with us. Again, this will be posted on our YouTube channel. We have plenty of more information on our website, and that information is on the chat. And we thank you for participating in this webinar. And please keep an eye for that email for the survey. We will really appreciate some feedback, and also just to gauge your interest into future workshops and also webinars. Thank you very much.
Thank you. Bye. Thanks a lot.
Thank you, everyone.
Thank you, Maya, for putting this together, and everyone.
Yeah, and just write if you have questions. Bye.
Thank you. Bye.